In commercial poultry farming, viral infections can lead to significant economic losses. If flocks are not sufficiently protected through vaccination, pathogens can spread rapidly among the birds. 3 very common viral infections and the corresponding highly contagious diseases in poultry are caused by the ND virus, triggering Newcastle disease, the ILT virus, which causes infectious laryngotracheitis, or the MD virus, responsible for Marek's disease. Live HVT virus vaccine offers effective protection against Marek's disease. The 2 viruses are very similar. However, in contrast to the MD virus, HVT is non-pathogenic in chickens. Vaccination with HVT triggers an immune response against MD virus. This includes the activation of macrophages that phagocytose the virus. Other cellular immune processes involve natural killer cells, which destroy cells already infected. Within 2 weeks, the vaccination also leads to a production of antibodies against HVT surface proteins, as well as against those of the similar MD virus. In addition to these useful properties, HVT also makes an excellent starting point for the production of combined recombinant vaccines that are effective against more than just one disease. Recombinant vaccines consist of a vector, such as HVT, expressing foreign antigens. HVT is very suitable as a vector because it has a relatively large genome that enables the insertion of foreign genes. On our way to understanding recombinant vaccines, let's first take a closer look at the mode of infection of the ND virus causing Newcastle disease. To achieve infection, it is indispensable for the virus to attach itself to cells in the bird's body. The fusion of the ND virus and the chicken cell membrane is mediated by the F surface protein. The virus now releases its nucleocapsid into the cell, starting the infection process. Once infected, new virus particles are produced and the cell is destroyed. Blocking this fusion protein is sufficient to prevent an infection with the ND virus. The idea behind the recombinant vaccine is to insert the fusion protein gene into the HVT vector. In a first step, the single-stranded RNA coding for the F protein of the ND virus is isolated. But HVT, the vector, contains double-stranded DNA. Therefore, transcription of the isolated RNA is necessary via the complementary single-stranded DNA into insertable double-stranded DNA. A sequence of non-essential DNA from the HVT is multiplied and then isolated. After adding a promoter and a terminator region, necessary for initiation and stopping of transcription, the F gene can be inserted into the vector. Following multiplication of this newly formed DNA sequence, the non-essential part is recognized and then integrated into the HVT genome. Recombinant HVT is now cultivated in cell cultures to provide large amounts of the recombinant vaccine.